Hello everyone, welcome to Pearson Bio Academy. If you are new to my channel, this is Fazna Imran and this is the place where you can top up your biology knowledge with our past paper explanation through very short and clear video series. Let's get diving. Today we'll be focusing on Pearson Edexcel International Advanced Level Paper Reference WBI 1101 Unit 1 Molecules, Diet, Transport and Health. So this is the course outline. If you have watched my all video series, then you can recognize. So this video is going to cover the chemistry of life. And under chemistry of life, we have carbohydrates, chemistry of water, proteins, lipids, and enzymes. And today we will be tackling questions on proteins. And we'll look questions from 2020 to 2024. So amino acids are the monomers of a protein, the building blocks. First, we will tackle the structure of amino acids. There is a carbon at the center and it's attached to a carboxyl group, a hydrogen atom, an amino group, and finally an R group or side chain. Now if we look at an amino acid, these three parts, the carboxyl group, hydrogen, and amino group are the same for every amino acids. Only the R group or side chain differs among them. Here we go with the first question. It's from the 2022 June paper. Now this question is part of a larger question and we will tackle the complete question later. For now, let's focus on this specific part. So in this question, you are asked to draw the complete structure of an amino acid. So the context provided mention that keratin contains a high proportion of the amino acid cysteine and disulfide bridges form between the R groups of two cysteines, and the diagram shows the R group of cysteine. So the key idea here is that they might provide the R group and ask you to complete the structure of amino acid. Therefore, it's important to understand and memorize the common parts of an amino acid structure. Let's break it down step by step. First, at the center you have a carbon atom. Attached to this carbon, a hydrogen atom, a carboxyl group, an amine group, and finally the R group, which can vary depending on the amino acid. Now let's tackle the characteristics of R groups and their importance. So some R groups of amino acids are hydrophobic or nonpolar. Let's take an example. Leucine. In leucine, you will find the three common parts of an amino acid, and in addition, the R groups contain hydrophobic or non-polar component. We can explore more examples of amino acids with hydrophobic R groups. For instance, alanine, valine, tryptophan. So these are just few examples where the R groups exhibit nonpolar or hydrophobic properties. So understanding these characteristics is essential because the hydrophobicity of these R groups play a significant role in protein structure and function. And the next important property is that some R groups are hydrophilic or polar, which is the opposite of the previous ones we discussed. For example, if we take serine, it has the common structure of an amino acid. But in addition, its R group includes a hydrophilic polar group. And other examples include asparagine, cysteine, and tyrosine. So if we look closely at each of these amino acids, you will notice their R groups contain a polygroup, making them hydrophilic. And the next property is sulfur containing amino acids. So among the 20 amino acids, only one, cysteine, contains sulfur in its R group. And this unique property makes them particularly important for the function and structure of proteins. Let's discuss in detail later. And now let's address this question: what are the importance of R groups? So the very first point is that R groups determine whether an amino acid is hydrophobic or hydrophilic. And this property directly influences how amino acids interact with each other and their surroundings, which ultimately affects the folding and overall structure of proteins. So in the diagram, you can see a sequence of amino acids represented in red and blue. So the red amino acids, they have hydrophobic or non-polar R groups, and while the blue ones have hydrophilic or polar R groups. So initially in the primary structure, these amino acids are arranged in a linear sequence. However, when the protein falls into its 3D structure, interactions with the aqueous environment come into play. So the hydrophilic or the blue R groups are positioned on the outer side of the protein, facing the surrounding water, while the hydrophobic R groups or the red color one are tucked into the core of the protein. And this arrangement is crucial for maintaining the protein's stability and proper function. Right, so let's quickly go through the question. It's from January 2021 paper. The question states that the role of hemoglobin is to transport oxygen and carbon dioxide in the blood. And the diagram shows the structure of adult hemoglobin. And you can see that P is indicating a core part of this chain or of this subunit, and Q is denoting some part of this polypeptide chain. And I think you already know the structure of hemoglobin. It's simply made up of polypeptides, and also it has two types of subunits called alpha and beta, and each subunit has a heme group at its core. So here we go with the question. Explain the properties of amino acids located on the outer surface of the hemoglobin, for example at position Q. So the question mainly targets you: what characteristics should the amino acids have to be located on the outside of the hemoglobin molecule? So let's look at the RBC. This is an RBC, and inside the RBC you can see a cross section. If you look closely at the cross section, so this is how the hemoglobin molecules are tightly packed inside the RBC. You can see that red part represents the hemoglobin, while the surrounding blue part is the cytoplasm of the RBC, which consists of more aqueous environment. So hemoglobin needs to function within this aqueous environment internally. So here is the hemoglobin molecule, and around it there are 4 molecules. Therefore, the outer surface of this hemoglobin molecule should be hydrophilic to interact with these H2O molecules. So I hope you understood the concept, the idea that the outer surface of these hemoglobin molecules should be hydrophilic. So therefore they should have R groups or side chains that are hydrophilic or polar. First, let's look at the keywords you need to use for this question so that you can construct your own answer. And the keywords are R, or these side chains are polar or hydrophilic group, and they tend hemoglobin molecules to be dissolved in the cytoplasm. So using these keywords, you can structure your answer. For example, you can write that the amino acids in the outer surface should have the R groups which are polar or hydrophilic. Then make sure to explain why this is important. You need to include the reason. This allows the hemoglobin molecule to dissolve in the cytoplasm of red blood cells. So that's why the outer surface needs to be hydrophilic. So if you give an answer like this, then it would be perfect for this question. So next we will move into what are the other important roles of R groups, and one of their key roles is the ability to form disulfide bonds. So the basic idea is that if amino acids want to form a disulfide bond, they need to contain sulfur. As you know, cysteine is the amino acids that has sulfur in its side groups. And in this diagram, the yellow represent cysteine molecules, and you can see how they form disulfide bonds between these sulfur atoms. And these disulfide bonds are specifically formed between two sulfur-containing R groups. And also, these disulfide bonds cause the straight polypeptide chains to bend, contributing the 3D structure of the protein. Let's begin with a question about the properties of R groups, which appeared in your past paper. So this question is from the June 2021 paper. It focuses on the structure of the cell membrane and how it influences its properties. The diagram provided shows part of a cell membrane with specific proteins and other labeled parts P, Q, R, S, T. However, I won't be discussing all the components of the membrane in this video. Instead, I will address those in a separate video series dedicated entirely to cell membranes. For now, we'll focus on the questions related to the protein in this diagram. The question asks, explain the role of primary structure in determining the properties of the protein labeled in the diagram. Here, the key target is to describe the primary structure, refers to the sequence of amino acids in the protein. Therefore, your focus should be on the characteristics of these amino acids. So this means that indirectly you will be discussing the properties of R groups attached to these amino acids. So let's get back to the diagram. So in this diagram, you can see the protein, half of these embedded within the fatty acid tails, and the other part is exposed to the aqueous environment of the cell, or possibly the outer part. So the key idea is here that the aqueous environment is hydrophilic, while the fatty acid region is hydrophobic. So this means that the amino acids facing the aqueous environment should have hydrophilic R groups, whereas the amino acids embedded within the fatty acid region should have hydrophobic R groups. So this distinction in R group properties is essential for the protein structure and function. Now let's look at how you can construct your answer using the key points. First, remember that the primary structure refers to the sequence of amino acids. And this sequence determines the position of amino acids, which in turn dictate the types of bonds that will form. In this case, the hydrophilic R groups are positioned on the part of the protein facing the aqueous environment, while the hydrophobic R groups are positioned within the phospholipid tails. To write your answer, start by stating that the 3D structures of the protein is primarily determined by the sequence of amino acids. The arrangement of amino acids dictates the formation of bonds between R groups based on the position of these groups. For instance, disulfide bonds. In the label protein, the amino acids which have the polar R groups or hydrophilic on the outside of the part of the protein facing the cytoplasm or facing the aqueous environment. The amino acids with non-polar R groups, hydrophobic on the outside of this protein that is embedded in the phospholipid tails. So far we have discussed about the structure of amino acid and the properties of R groups and how they impact in the protein structure and function. And also we discussed some past paper questions regarding the amino acids. I hope you understood the concept and thank you for watching my video. If you haven't subscribed to Pearson Bio Academy yet, please subscribe and hit the like button for this video. If you enjoyed this video, don't forget to hit the like button. If you haven't subscribed yet, please subscribe now and share this with your friends. Let's meet in the next video series, which will be about the protein part 2. Bye.